Noncoding RNAs often have multiple isoforms. In vitro overexpression studies, it is often difficult to study all these expressed isoforms. This technique allows users to drive expression directly from the genome and enable the cells to express the endogenous isoforms for a particular non-coding RNA. This powerful technique allows the user to specifically direct the expression of any gene in the genome with fidelity and accuracy. By designing guide RNAs to specific genes, one is able to use the endogenous gene splicing machinery to express the natural isoforms in a given cell. Demonstrating the procedure will be Robert Rankin, a postdoc from my laboratory.
To design the guide RNA sequence which is located within 100 base pairs of the transcriptional start site, use genetic databases such as BLAT to make sure that the guide RNA is unique to the gene of interest. Store the guide RNA in the deactivated Cas9 plasmids at 4 degrees Celsius. Streak out E. coli on a luria broth agar plate with ampicillin and grow the bacteria overnight at 37 degrees Celsius. The next day, pick a colony and grow the bacteria in 5 milliliters of LB with ampicillin overnight, vigorously shaking the tube in a 37 degrees Celsius incubator. The next day, add 2 milliliters of bacteria to 200 milliliters of LB with ampicillin in a 2-liter flask, vigorously shaking the flask overnight in a 37 degrees Celsius incubator. Spin down the bacteria at 3,724 times G for 20 minutes and proceed to DNA purification. To create the dCas9 containing lentivirus, first coat